Hello class, this is Mr. Alfonso. This is the first lesson in Unit 6. So here's our pacing schedule. The first thing is do now on Canvas. Next is notes, which is this video. Third, there is a worksheet and a reading that goes with it for you to complete. After that, answer the essential questions on the discussion post, and finally do your Mastery Connect. Before we continue our lesson, let's take a quick look at your essential questions. The first thing you need to look at is the topic today is DNA structure and replication. Now, the first question you need to answer later is what phase during the cell cycle does DNA replication happen in? And that's a good review. Actually, you might be able to answer that without going through this lesson, because that's a Unit 5 question. 2, what does the word semi-conservative mean? And 3, if I have 15% A, how much T, C, and G do I have? Again, you will answer all these questions by the end of the lesson. Now our focus on the topic is DNA structure, and if you look, Google the word DNA, take some time to do that. Tell me what names pop up, right? Pause real quick. Now, the names that you see are actually two males. It should be Watson and Crick. But one, however, they did not give credit to a female scientist who played a very important role in discovering what DNA looks like, something that is huge and part of biology. Her name is Rosalind Franklin. She basically did all the work for Watson and Crick, and despite her working very hard and actually dying for her work, she did not get credit. She did not get a prize. She did not get the money. So I want to take time here and commemorate Rosalind Franklin, because our female scientists don't get enough credit for what they do. She died from cancer, primarily just because she took X-rays of DNA, and the X-rays had caused her cells to be broken. Right? We talked about how the DNA, the cell cycle checkpoints got messed up, and that caused her cells to keep reproducing uncontrollably, which is cancer. So that's a quick nod to commemorate Rosalind Franklin for helping discover what DNA looks like. Now let's take an actual look at what DNA is, what the structure is. So first off, what is DNA? Go and get your notes, piece of paper, and pencil. DNA stands for deoxyribonucleic acid, and that's a mouthful. That is deoxyribonucleic acid. Okay? The D is deoxyribo, the N is nucleic, the A is acid. Alright, second, what does DNA look like? Now honestly, before we know what it looks like, you need to know what it's made of. DNA is made of 1 nucleotides, and we actually know nucleotides from Unit 2, and they're made of 3 things. Sugar, deoxyribose, they're made of phosphate, and they're made of nitrogen bases, which is ATCG. Now what you need to know is these always come in pairs. A will always go with T, and C will always go with G. Now how I like to remember this is apple turnovers, right? AT, and CG is going to be collard greens, CG, okay? Now the structure of DNA resembles, it's going to look like a staircase, right? A winding staircase or a twisted ladder. What we call that is a double helix. So the structure of DNA is called a double helix. Make sure you know that, say you get that word, double helix. Now there's 2 parts to it. One, the sides of the ladder, right? That's the backbone, and it's made of phosphate and sugar. That's important because that's what keeps it strong, right? That's what holds it up together. So the backbone of DNA is the phosphate and sugar backbone. While the middle part of the ladder, the rungs, is what we call nitrogen bases. That's the A, T, C, and G. Now one thing I realized, we didn't label what those A, T, G, and C means. Let's quickly discuss that. So A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. Make sure you record that. You will see those names again. Now what is the function of DNA? There are 3 things you need to know. 1, it is the blueprint for life. That means your DNA determines what you look like and everything else in your body. 2, determines heredity. That means That's what you're going to pass on to your kids and what you got from your parents. 3, code for making proteins. Now, proteins are important because you are mainly 98% protein, right? Your skin, your bones, your muscles, your eyes, those are all protein. And DNA is basically the instructions to make it. So we'll cover that further on in this unit. But let's still focus on DNA. Again, we talked about this. Common terms of DNA, you might have seen the word chromosome, the word gene, and the word trait. Trait is the newest one. And let's go review about... So let's go ahead and answer our essential question number 1. What is replication? Replication is when you make copies of DNA. And this is a quick review. Remember, replication occurs during the S phase of the cell cycle. G1 is where the cell grows. S phase, DNA is replicated, is copied. G2, it prepares for mitosis. And mitosis is where the cell divides, right? PMAT. Perfect. Moving on. Where and when does replication happen? Where? Because it works with DNA, DNA can be found in the nucleus in eukaryotes. But since prokaryotes don't have a nucleus, the DNA of prokaryotes will just be in the cytoplasm. And when does it happen? This happens all the time. All the time. Okay? Now, which is a quick activity. DNA is the first picture. Over here is going to be our chromosome, right? It looks like a straight line. Then we have trait. That means a trait is what you look like. And the gene is a small part of DNA. Let's keep going. I wish we were in class because then this would be a good chance to get some candy. Basically, the question is what happens if a cell does not replicate its DNA before the cell division? And that means the cell will not have enough DNA, which will cause a lot of issues. Okay, and this is more for those who are interested. If you don't want to watch this, go and skip ahead after I go over the slide. But this is for more of my students who want to pursue biology in college. Now, how DNA is replicated is DNA, like this letter, it's going to be unzipped, right? It's going to be unzipped, something called DNA helicase, which is, it ends in A, so it's an enzyme. So it unzips, and what happens is each part gets a new copy. So we'll see what more of that means later, but basically, you go from one DNA, and now instead of, you're going to make 3 different, now you have 3 sets of DNA. We'll talk about how that looks later. I'm sorry, 2 sets of DNA. So results of DNA replication is this. We have 2 identical DNA molecules that are produced during replication. Like I said, it unzips and new ones are added. These new strands are semi-conservative, meaning DNA is made of 1 old strand and 1 new strand. So if you look at the picture here, the red is the old strand of DNA. And if you look in number 2, they unzipped and they added the new strand, which is blue. So every piece of DNA is made from 1 old and 1 new strand. That's why we call it semi-conservative. Half old, half new. Now let's take a look at examples of DNA replication, right? This is going to be a little confusing, but it'll be very easy once you understand it. So here's an example of DNA. AAA, TTT, CCC, and GGG. Now remember what I said earlier. Apple turnovers and collard greens. A's, when you replicate DNA, you're not copying it exactly. You're going to write down the nitrogen base that goes with it. So T's, A's go with T's, T's go with A's, C's go with G's, and G's go with C's. These letters are known as nitrogen bases, or nitrogenous bases. Okay? Now, look, this one, A goes with T, so if T and T goes with A, so TAA, TCA, AAA, and CCG. It's a really mix and match pattern. You just need to know A goes with T, T goes with A, C goes with G, and G goes with C. Let's look at a couple more examples just to help you get it. AT, TA, 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 CG, CG, GC, GCAT. Okay? If you have any questions, please come during my office hours and I'll be more than happy to explain this to you further. Now, is this one correct? Let's see if we can spot where something is wrong. Do you see the error? Right. Where in the yellow, it says TAG, it actually should be TGG, because we know that C should go with G. And another error here is TGA, that should actually be TAA, because we know that T goes with A, right? We have all these errors here. And in the final one, there is another error. A does not go with A. A goes with T. So make sure you stay aware and look at what goes on. Now I know this is a long lesson, but we're getting to our final thing, something called Chargaff's rule. So it's math, so bear with me. So the amount of DNA you have will always equal 100%. So that means the amount of A's plus the amount of T's plus the amount of C's plus the amount of G's you have is 100%. And we know A is T, so the amount of A will always equal T, and the amount of C will always equal G. Okay? When we'll do more practice with this later, during our office hours, and I'll actually upload an additional video. I don't want to overwhelm you with this source of knowledge, but that is where we're going to stop. Make sure you got your summary, and go ahead and look out for the video part 2. Alright, y'all have a great day.